He was on the state CMPS horticulture and education committees for 10 years. And as I read these, I want you to know that these quick little things embody huge amounts of work and very influential work. So for example, on the state CMPS horticulture committee, when he was on that, back then, all the experts said, oh, you can't make a simple little brochure on the benefits and what to do about gardening with natives California. There's just so much variety and stuff. So here in Orange County chapter, we put together one that was certainly good for our chapter and that we thought could be raised above. And a lot of that was with guidance with Dan. Then he walked it right up to the state committee, dropped that on the table and said, I think we can. They agreed, and then they put state funds to increasing the graphics. And that actually went through 2 to 3 generations of each time adding more information, getting bigger, because they're right, there's a lot to know, and then adding more beautiful graphics. And that's just one of the things that he could do. So he could walk in, people know he knows what he's talking about, and he just calmly get things going. So as we read through all these others, know that that simple little word I say probably embodies quite a bit of work. In our chapter, he was an initiator or early driving force for our chapter plant sales, our book sales, our chapter support for the book Wildflowers of Orange County and the Santa Ana Mountains, which obviously Bob Allen and Fred Roberts put tons of work into. But as far as making it come about with some of the legal contracts and getting people to read to and edits, Dan was a big player. Some of our early lawsuits, the Invasive Exotics Committee in our chapter, the Horticulture Committee in our chapter, the grants, the symposiums and workshops, lots and lots of work by Dan. In our public programs, if you've been watching them, he was bringing us experts like Bart O'Brien, Carol Bornstein, John Keeley, Greg Rubin, Naomi Fraga, and Reagan Dunn. And he's been doing that decade after decade. And like I said, I think we've said before, Reagan Dunn was the one we saw early this year. And then half a year later, her research, combined research with other people, was in the journal Nature, the very top journal Nature. Also, we recognize Dan partially because of his recognition of all the other people who had been working. He'd do awards, articles, verbal kudos, and support of people. Wonderful guy to work with or under side. We enjoyed having him as a speaker, as did other organizations. And then at booths and tables, he'd be an expert. And I want to say something about that. We would, a bunch of us would go, like when there used to be the Fullerton Arboretum green scene, it was great to have Dan there because the people would come and they'd ask us questions and we'd have a 2 or 3 double wide, booth. And we'd be answering these questions, helping people out. And then of course there'd be someone who comes out with a question that's just way above us. And so we'd look to the side and you go, see that guy over there? That's Dan Songster. Occasionally it would be Celia Kuchar also. And you see that line behind him? Please get in that line because he's the guy who can handle it. And then it's kind of a joke, I'm sorry to say, we'd also put other people in those lines. People would come to us and say, okay, I saw this plant the other week. It's white and it's something like about this size and it has green leaves. Do you know what I'm talking about? And we just go talk to Dan because Dan just had a way of pulling more out of them and getting to where they wanted to be, or somehow else giving them useful information when the rest of us are going, oh man. Okay, anyway, he's also known for creating handouts and newsletter articles, including the long-running Native Gardener's Corner, which has that focus on a question, and then he gets experts and members to reply. And I have a little smiley face here because that will continue. Don't worry, he's not completely gone. And I'm sure we will see him and rope him in to other things too. But for when he hopefully watches this recording, I'd like everyone to give him a clap of hands, or if you just have a glass of water, a public cheer and thank you to Dan for decades of just wonderful work for the chapter and a great guy to work with. Thank you.
Cheers. You can't see my water bottle. Cheers to you, Dan. Thank you for all your work.
With that, unless any board members have some specific high need announcements. If you do, speak up. Otherwise, we will go to the main event tonight.
Okay.
Honey, take it away.
I'll take it away, yeah. Unless there's any other announcements. So today we have two wonderful speakers. Let me see if we can get them set up. I guess Laurel, if you can start sharing. So every year, annually, we have the Charlie O'Neill Grant that are awarded to graduate and undergraduate students that are planning to conduct field research related to biology, ecology, floristics, taxonomy, and ethnobotany of native plants. And our two speakers here today, Laurel Sebastian and Rachel Tageant, they were awarded the full amount last year out of all 6 awardees, actually. And without further ado, let me introduce Laurel. I'm gonna read my spiel. So it's fresh off the press too. We got some new updates. So Laurel recently received her master's degree in ecology in Jennifer Funk's labs at UC Davis. So Jennifer Funk used to be down here in Orange County at Chapman or before. And Laurel is currently working on publishing her thesis work and recently accepted a position as a restoration ecologist for River Partners. Laurel is originally from Santa Rosa, California, and Laurel spent 5 years doing field research across diverse ecosystems of Colorado, Washington, and California before returning for grad school. And without further ado, Laurel's going to talk about her presentation.
Great, thank you. Yeah, thank you for all having me tonight and also funding this research. As you can see on the screen, hopefully, I will be talking about plant traits and diversity and how the environment all shape soil carbon dynamics in some restored grassland plots down in Irvine. And yeah, as you said, I was in the Funk Lab and just graduated, so happy to kind of start to talk about this research in the past tense. Let's see, I'll make sure I can go to the next screen. Okay, so, just the broadest level background, as we all know, there's really a need for some targeted restoration across our state and across the planet. But that's really because we have this global loss of biodiversity, unfortunately, with pressures like climate change and habitat loss. And that's a problem for many reasons. Of course, we love all these plants, but also because biodiversity is known to be really important for enhancing ecosystem function. So basically, as this graph is one of many that kind of shows on the bottom axis there, if you have more plants, you tend to get more benefits from the ecosystem, like that's showing resistance to drought, but that's also true for kind of closer to the research I did, true for carbon sequestration into the soil, supporting pollinators, all sorts of things. So we know we need to restore this biodiversity, and that generally can take 2 different approaches. So one is to just generally aim to restore biodiversity, scattering a diverse seed mix with hopes that you'll get a diverse plant community. We can also try to target functional traits and to get a really specific type of diversity. So functional traits are measurable plant characteristics. So, you can measure the depth of roots or the density of roots or the thickness of a leaf, and that can tell you a little bit about what type of plant that is and what role it plays in the ecosystem to help us meet specific management goals. So for my research, I'm interested in particular to try to kind of tie that to soil carbon storage and sequestration in grasslands. And that's important. I mean, all ecosystems are important, but I think grasslands are interesting in that they are one of the largest terrestrial carbon pools. And that's surprising for a lot of people because the carbon is underground. There's only so much biomass above ground, but underground, these grasses, the roots, all the litter over time can accumulate to really have a lot of carbon. And then, I'm doing this research because there really are still a lot of unknowns about how plants and the traits of those plants, their functional traits and environmental variables all interact to affect soil carbon. So, my goal here is to try to inform research and restoration strategies to maintain and increase soil carbon over time. That kind of fits into some potentially carbon markets, but as just general ideas of restoring soil carbon. This will start to get a little bit detailed, but I'll try to, remind you, and you don't have to remember all of these things as we go along. But I decided to measure soil carbon in 5 different ways. At a broadest level, plants are absorbing carbon dioxide and building structures made out of carbon, and that can end up in the soil in a couple different pools. Total carbon is just the sum of all the different types of carbon in the soil. So you don't need to know much more than that. It's kind of the simplest way to measure it. But we can start to look at these sub pools too, to kind of tell us like, where did this carbon come from and how are plants influencing it? One of which is particular organic carbon, and that's just plant detritus, like roots and leaves ending up in the soil. It decomposes over the time, but it also is an important component of soil carbon. We also have water extractable carbon, which is a pretty small percentage, but it can be really important. It's basically the exudates or the sugars leaking out of roots into the soil. And the sugars that plants are making during photosynthesis, a lot of those end up exuding into the soil and feeding microbial growth in a lot of the soil community. Then relatedly, I measured microbial biomass. So there's kind of a way to get an estimate of how rich the microbial community is in the soil. And that's really important because the more we are learning, the more we understand that microbes are essential to kind of transforming carbon from plants and stabilizing it into the soil long term. And where it kind of ends up, this most stable pool is called minerally associated carbon. And that's because as the kind of microbes and different soil processes process some of that carbon and it breaks down, some of it gets bonded onto minerals in the soil, like all of our silts and clays in the soil. And it can be a really stable place for it to be. It varies depending on the soil, but like it can be in that pool for thousands of years. However, we'll get into this later, but that pool can saturate, so it can kind of get full. You can think of the minerals not really being able to like have any more space to bond to that carbon. So we'll get more into those carbon pools later. And I have one more complex side, and then hopefully I'll kind of break things down as we go. And this is just kind of another tricky background for those of you who don't have this kind of experience thinking about traits. A lot of the science breaks it down into these kind of gradients. So, we can measure anything about a plant and put that along some kind of axis or gradient of its traits. So the most essential one that people that do this kind of research talk about all the time is this resource conservation gradient, where we have kind of slow plants and fast plants. And the slow plants might have thick tissues that really resist decomposition. You can kind of think of like a valley oak leaf or something. It's a little bit thicker, and it's going to linger around in the soil longer than some really fast growing annual weed that has a nice soft leaf and kind of decomposes quickly. And those kind of feed different potential carbon pools. So we're kind of interested in what about the plants is feeding these different pools. And then also there's a collaboration gradient, which is actually pretty new. Just in the last couple of years, scientists are figuring out that this is probably a really important way to describe plants. And that's this purple gradient there, or kind of axis, where we have on one side these plants that tend to outsource their resource acquisition in the soil. So some that have really thick roots, and those thick roots actually allow them to host like fungi, mycorrhizal networks, that they kind of outsource that work to the networks to get nutrients and water from the soil. And again, because those are kind of thicker tissues, they might end up in our particulate organic carbon pool, like that carbon pool that's made of plant detritus. On the other side, we have these DIY or do-it-yourself roots, where those are really thin and numerous, maybe like kind of fibrous grass roots that are kind of throughout all the soil and are able to kind of access all the water and nutrients that a plant needs itself. So we'll talk about those in my results. And then the other thing I was interested in is environmental factors. So, with climate change, a lot of researchers are thinking about this kind of thing where, precipitation is maybe going to change more and more with our future. And that could be important because drought can decrease plant growth, and that could then decrease how much is getting put into the soil or can affect decomposition. And then also things like land use history was included in my study, because whether a site was tilled as farmland in the past, or whether it's been native grassland for decades, is gonna affect how much carbon is already in the soil, and that can affect how much can kind of build in the future as well, in some cases. So now, kind of starting to get into my particular experiment, we had 210 plots, and those are each of the smaller black squares. Each plot got seeded with 10 of 35 species. So each plot was a little bit different in its composition. And then those plots were kind of grouped together into different precipitation treatments. So they either got 50% or 125% of the annual kind of average California precipitation, or at least for Southern California. So kind of dry plots and wet plots. And those are kind of each outlined in those light or dark blue squares. And those were under, you can see it in the picture, kind of there are these hoop houses that could exclude rain and irrigate it after. And then we also had this land use history treatment, which is a little bit tricky to explain. It's basically, there were different experiments on these plots prior to our experiment that seemed in the end actually matter a lot. So in the light green box, those plots were tilled for longer and cover cropped, and they were more similarly treated to like croplands. And then in the dark green box, those plots had native shrubs grown out, including like California sage and some other shrubs, for 6 years before our experiment began. And that seemed to have a really important carbon signature. And so we'll get into why that mattered a little bit for our results. And so I went over 2 years to collect soil, and generally collected from the first 15 centimeters in the soil with the soil probe. While we were there, we surveyed each plot to get an idea of exactly what ratio of plants was in each plot. And that allowed us to calculate trait values. So like on average, how tall are the plants in this plot? What's their root structure like? And do a little bit of simple math there to get some idea of their traits. And then we sieved and processed all that soil from the 210 plots in 5 different ways to get 5 different carbon readings. And then the results you'll see today are just kind of linear regressions between the plants and the soil. So my research questions that I'll get into now are: how do environmental variables, plant diversity, and functional traits affect soil carbon? And do those plant-soil interactions vary with precipitation and land use history? So like, how are the environmental variables affecting the plant-soil interactions? I also did look in my thesis at time lags and like how long it took these effects to show up, but we're going to skip over that today for the sake of time. So yeah, looking at kind of the broadest level results, I did see that land use and precipitation affected our carbon pools. So in these first 4 graphs over here, generally the dark green bars are higher than the light green, and that's showing us that that land use history did have an important effect on the carbon in our plots. So those plots that had more shrubs growing for longer and they weren't disturbed every year had a lot more carbon versus the ones that were tilled every year and planted with cover crops for longer. And I thought that was interesting, kind of from the broadest level, and just in that our management differences seem to really have lasting effects. It's only 4 years, but 4 years after, by the time my experiment ended, I was still seeing this strong signature 4 years after there were any shrubs or any tilling happening. And because all of my plots were tilled, it doesn't seem that tilling kind of erased all progress, because there's a lot of discussion these days about like, oh, well, what if you restore a grassland and then and it builds a ton of carbon, and then you have to till it for some reason. Is it all just going to disappear instantly? But that doesn't seem to be the case, that the signature really lasted many years even after tilling it, which is, you know, good news, that our management makes a difference. And then also, I only saw, this is somewhat surprising down here, we only saw one pool, which is our minerally associated carbon, showed any response to the precipitation treatment, which, yeah, we thought maybe precipitation would matter more, but it was in some ways good news, because maybe with, you know, because we're going to see more variation in our precipitation, maybe that won't have a huge effect on all these pools. Maybe they're more stable than we think, with a little bit of variation in precipitation from about 50 to 125%, didn't seem to have a huge difference overall in these pools. And now I'm going to get into a little bit trickier results. I'll only show you 2 slides that are like this, because this is kind of what most of my results in the study were like. They were complicated and a little messy, but that's kind of how it seems like plant-soil interactions are often a little complicated like that. So in this first result, I found that species diversity was beneficial for microbial biomass and that really stable pool, which is minerally associated carbon. But I only really saw that effect in wet conditions. And so starting to break that down, so the top, let's see, these top two graphs are showing that, you know, there is this slight positive correlation between species evenness, which is kind of a way to measure species diversity, and these 2 pools, but they're only it's only positive in those, yeah, dark blue bars that are from the wetter plots. That seems to be, well, I guess I saw the same effect also show up for microbial biomass, so the amount of microbes in the soil, with that fast, slow gradient that I showed you in the beginning. So whether like plants really linger around for a long time or whether they're really biodegradable, it showed that there was kind of this positive correlation with those labile, like fast traits of plants. So maybe you can think of a weedy annual, like or a mustard leaf or something that decomposes really readily. But I thought that maybe that's only happening in the wetter plots, because basically that's the amount of water the microbes need to survive and thrive. And when they're well watered, those labile kind of easily decomposable tissues that might have a lot of nitrogen and yummy things for the microbes to eat are stimulating the microbes. And that's the same for diverse plant tissues. So you can imagine a bunch of different types of plants would feed more types of microbes if at least there's enough water for them to survive. But we see the trend is the other way in the dry plots, and that might just be under a little bit of drought stress. The microbes aren't doing as well, and maybe diverse and those kind of acquisitive or fast plants may actually be outcompeting the microbes for water a little bit when they're under drought stress. The last point I wanted to make was that we did see the same trend here between microbial biomass on the left and minerally associated organic carbon on the right. And that mirrors a lot of research kind of showing back to our different carbon pools. That mirrors research that shows that microbes are important for making minerally associated organic carbon. So maybe the litter and the roots, these things that are feeding the microbes, are ending up also in the minerally associated pool at the bottom right here, which is kind of our most interesting pool, and that's a really stable place for carbon dioxide and carbon to end up long term. I have one more result here. I guess I didn't do a great job explaining this, or I didn't try at all to explain this graph on the bottom right. It's a little bit confusing if you haven't seen this type of plot before, but these are all the traits I measured, and they kind of, when you put them all on the same graph at the same time, the computer kind of figures out which ones align with each other. And so the kind of code I ran figured out that these traits all kind of run along the same axis, and that means they're all kind of related. So over here, I know that, you know, nitrogen, which is this N here, is like a fast trait, and then leaf mass area over here is a slow trait. It's like those thicker, heavier leaves that take a while to decompose. And then same for the second axis, things generally align where you have like high root diameter on one side and then another trait that's kind of opposite on the other side. So you'll see that again on this. So now I want to focus, instead of on the fast, slow gradient, we're talking about the collaboration gradient, or the one that's more about whether they have thick roots or thin roots. And in this result, I found that thin roots, or these kind of DIY or do-it-yourself roots that are dispersed throughout the soil, tend to boost minerally associated organic carbon in low conditions. So that's kind of shown. It's a little bit tricky, but this collaboration gradient in the negative values over here are like, this is specific root length. So that means how many roots are in the soil. When that value is high, we're kind of looking at this end of the graph. So we have higher minerally associated organic carbon, but only in these plots that have low carbon in general. And those are the plots that have been tilled for longer. So it seems, my main theory for this, of why this is happening, is that thin roots are actually better at exuding carbon directly into the soil and forming minerally associated organic carbon. So I'll show you this again, where it seems like you could imagine, you know, here's an example of a root, and the thinner the roots are, and the more dispersed they are throughout the soil, it seems like more carbon is ending up directly in this minerally associated organic carbon pool here. So kind of following this arrow, and meaning the, you know, sugars that are coming out of the roots are bonding right onto the minerals in the soil, which is a great stable place for them to be long term. And then I added this red X here because it doesn't always seem to happen. Again, we have this kind of flatter line here where we see a flat or even opposite response in the plots that had the land use history with more shrubs for more time, kind of showing, and had more carbon overall. And in the very beginning, I mentioned the only problem with minerally associated organic matter is that, you know, it's the most stable place for carbon to be long term. It's a great pool where we want our carbon to end up, but the problem is that it can saturate. It can basically kind of become full, and it's at least slower in making these bonds between carbon and the minerals in the soil. And maybe that's what happened in our plots that already had shrubs growing for several years. They had a lot of carbon already in the soil. We're not seeing the same response there. So I want to kind of start to wrap it up for questions. I know if you're not thinking about carbon every day and not thinking about traits every day, that might be a little bit of a whirlwind, but just bigger pictures is that I, would recommend that researchers really try to plan for this complexity. So plant diversity and trait effects on carbon, I found, are really context dependent, and they also depend on the carbon pool I'm looking at. So like a lot of the results, I only saw, a certain response for a certain pool in a certain condition, which makes it a little bit hard to talk about my takeaways, but also, interesting in that I think not a lot of research does this. Not a lot of research breaks the carbon into 4 different pools, but I think it starts to tell us a lot more about how exactly are these plants affecting their environment or interacting with the soil. What about them makes them build soil makes them build soil carbon over time. And then some broader kind of management conclusions I was thinking about, one of which is land use history. Like I said earlier, it does seem like the prior management decisions we make really do endure for years, which, as long as we're making good, decisions in our gardens and in our parks and open spaces, I think, investing in our soil carbon right now is probably, I'm optimistic, like going to generally stick around. Even when we had one kind of tilling event in our plots, all that carbon didn't just poof into the atmosphere like some people claim it's going to. So I think, investing in our native ecosystems is a really great bet for long-term carbon storage. And then also considering your site history and precipitation can be important. It's a little more tricky to plan around, but just that carbon might accrue at different rates and different conditions. So it's not always going to be super easy to find like the one sure bet of the one plant that's always going to work everywhere, the one trait. But looking at the traits, I did kind of find that all the traits, the trait axes that scientists tend to talk about. So this resource conservation gradient, so whether it's fast or slow, and also that root collaboration gradient, so how kind of thick the roots were both important for predicting how much carbon is getting accumulating into the soil. And so, yeah, I guess I kind of already said that, but just that the acquisitive, fast-growing plants, I saw some evidence of those storing carbon into the minerally associated organic carbon pool and the microbial pool, which is somewhat new research. Like traditionally, people were only thinking that like the really slow perennials were the only way to go for making soil carbon. But I saw this result that even our kind of weedy, lovely annual wildflowers, things that grow more quickly, are also doing their part in sequestering carbon. And then that those kind of do-it-yourself root traits, the thin, abundant roots, seemed important for transferring carbon directly into that water extractable pool and the minerally associated pool. And then lastly, I just wanted to close on the idea that managing for biodiversity is always good. I didn't get too much into my, I have some other biodiversity kind of results, but just generally research over the last many decades has supported the benefits of diversity for soil carbon and so many other things. So even though I only saw some positive effects in certain conditions, it was a relatively short study. And if you're ever in doubt, just the more plants you can plant in your garden or if in an area you're responsible for kind of managing or restoring is a really safe bet for kind of feeding our carbon pools over time and sequestering soil carbon. With that, I'm sure I'm kind of at or over time. So I just want to kind of acknowledge that I had advisors and student assistants and a lot of research that helped me process all my soils, because it was a little bit more expensive than I thought to kind of send stuff off to the lab to get some readings back. So thank you all for funding it, and I will take any questions. If I can see my participants again, just a second.
It looks like there's a question in the chat. Ron is asking, do you look at soil texture and any correlation to carbon storage? It would seem that MOAC would be significantly higher in clay soils due to the higher surface area. Native grasslands are often associated with clay soils.
Yeah, that's a good point. I didn't look at soil texture much because they were all in the same area. Our plots were in it was at the South Coast Research and Extension Center in Irvine. I don't think I mentioned that, but I think it was like a 30-meter by 30-meter area or something. So we didn't have a ton of variation, and my soils were generally sandy. So that's one reason I maybe was starting to see like potentially indications that the minerally associated organic carbon was saturating, because they're sandy soils. And you're right, clays they have more potential to bond with carbon and therefore can hold more in that pool. But mine might have like been saturating earlier because they're mostly really sandy in that it's kind of a rare soil type for that area.
Any other questions? I was curious, like, I know a lot of areas in like California are subjected to nitrogen deposition, which can increase the resource levels on the environment, and a lot of times can lead to a lot of invasion occurring. In those areas that are heavily invaded and have a lot more inputs of nitrogen, how would you like what's your expectation of what the carbon pools would be like
Mm.
subterranean?
Yeah, I think it can be tricky because there are these like there can be these complex feedbacks where like depending on the carbon to nitrogen ratio, it can kind of change the microbial makeup, and sometimes that can occasionally lead microbes to kind of like stimulate the decomposition of carbon from the soil and the net release. But I think generally I would predict that, I mean, I'm sure there's research on this somewhere, but I think it would fit with the idea that legumes are generally good for sequestering carbon because they supply nitrogen to plants, and then the plants in their community, all their neighbors, can use that carbon or use that nitrogen to grow bigger. And then that's just a net more biomass that ends up in the soil. You know, more roots, more leaves, and then like over time more is accumulating carbon in the soil. So I would think it would generally have a similar effect. Yeah, unless you get like maybe too much. And then I know sometimes if you get nitrogen deposition, yeah, you have a decrease in biodiversity, which would probably have the opposite effect, where if you have, yeah, less biodiversity, you might have less carbon storage.
I was curious about the microbial biomass. Did you, by any chance, did you parse out if between like fungal and bacterial biomass, or would you expect there to be more of one community,
Mm.
depending on, I guess, vegetation then, huh?
Yeah, I didn't look at that for my research. I know a lot of people do, but yeah, it felt a little daunting to start to look at like all the different groups in the soil. So yeah, I guess I don't have a great answer. I never had the chance to look at it, but it was definitely fun to measure. You kind of take two pools of soil, and well, a little bit dangerous, I guess, but you do a chloroform fumigation and it like explodes all the cells of the microbes of half the pool, and then you take a normal sample of soil and like basically extract it and compare the two differences. So you have one that like there should be a big spike in carbon that you can read when you basically explode all the cells and the microbes in the soil. So it was kind of a more fun sciencey process to get, but that's as far as I went. I didn't key out all the different microbes and stuff.
Sure. I actually did a lot of chloroform fumigations in undergrad for a study on carbon dynamics in the desert.
Cool.
Yeah.
Yeah, it was fun. In my prior field work, I had just been like a field scientist. And so that was the first time I was working under a fume hood and wearing all my lab coats and stuff. So it was fun too.
Brad, do you have a question? No? All right.
Looks good. This looks like a good first half.
Yeah. Oh, I have one more question, I guess. I was curious about the role of fire. So a lot of times fire can like stimulate growth of a lot of our native plants, especially in like grassland systems if it's within a particular like fire return interval, right? How do you see that the role of fire playing in carbon sequestration? Because like you'd think like above ground the litter is just going to burn away, but it could potentially, encourage the growth of the plant, and then below ground would it be having more biomass and sequestering more over time? Like is there like a net long-term benefit of the fire potentially?
Yeah, I think, yeah, I would say, I haven't done a ton of research on that, but I think depending on the system, I could see that being a benefit. I could imagine like Stipa pulchra, I think, or a lot of bunch grasses like benefit from fire. And then instead of kind of dying out slowly, they would have a more vigorous growth after all of that like litter is really allowing them to photosynthesize again and stuff. So which would then probably have more root activity and allow them to kind of use their roots to sequester more carbon. And then the last thing I would say is I think that's a good reason to focus on grasslands for carbon, is that so much of the carbon is below ground or like biomass is below ground in the root system, that it's like a safer place for the carbon to be, versus in trees, unfortunately, can really burn on large scales and release a lot of that carbon.
Okay, cool. Thank you.
Yeah, thank you.
Alrighty. Yeah, of course. I think we are just out of time for questions now. I think we'll move on to Rachel. Thank you again, Laurel, and happy holidays. If you have to leave early, that's all right. So let me introduce Rachel. Rachel, are you able to share your screen?
Yeah.
Okay, perfect. I will stop. Cool. Okay. Rachel is a master's student at the Claremont Graduate University working out of the California Botanic Garden. Prior to attending grad school, Rachel received her associate's degree from Grays Harbor Community College and a bachelor's in biology from Washington State University. During her undergraduate career, she worked in the Marion Ownby Herbarium, where she conducted a floristic study in southeastern Washington and worked as a natural resource technician for the Washington Department of Fish and Wildlife. And Rachel's going to be talking today about her thesis about a floristic inventory of the Owens River Headwater area. Take it away.
Alrighty, thank you for that introduction, and I'm so happy to be presenting my research tonight. Yeah, so let's get started. So for those of us not completely aware of what a floristic inventory is, a floristic inventory is a systematic inventory which is important data to collect for land managers, future research, and other people interested in the botany field because it provides a foundational botanical knowledge of the area that you are studying in. So this research is a voucher-based research, and it creates a checklist of plant species with referenceable collections, and it essentially takes a snapshot in time of the plants that occurred in that area at that time. So this data can be used to aid or influence future research questions. It can be used by land managers to identify species of conservation concern or make land management decisions to avoid damage to ecologically sensitive areas or rare plants. Now I'm going to introduce my study area. So my study area is approximately 53 square miles. It is located, or as I like to say, sandwiched between June Lake on the northern end of my study area and Mammoth Lakes on the southern end of my study area. And to put that into a better context, it is about 43 miles northeast of Bishop, and it is about, again, 43 miles west of the White Mountains as the crow flies, and then we are about 300 miles north of Los Angeles. And for an even broader context, the purple circle in the map of California is the general area of where my study area is. And then my study area occurs on the eastern side of the Sierra Nevada crest, and the elevation ranges between 7,300 feet to 11,600 feet. So the climate here in my study area is a montane Mediterranean climate. So that means it is relatively warm, dry summers and mild, wet winters. However, compared to the rest of the eastern Sierra Nevadas, my study site gets more precipitation in the form of snow and summer showers because it is at a low point in the Sierra Nevada, and this is why Mammoth Mountain and June Mountain can be ski mountains. So the average high temp over the year is 56 degrees, where the average low is 29 degrees. However, the winters can get down to the 10s, and the summers can reach as high as 90 degrees. Luckily for me, Mammoth Mountain tracks a lot of snowfall. So historically, the snowfall of the season would be between 300 inches to 400 inches of snow. However, this past season, as many of us know, was an unusually cold and wet winter. So they had record snowfall of all time. So at the summit of Mammoth Mountain, they recorded a whopping 900 inches of snow, and down at the main lodge was 700 inches. And the Mammoth Mountain was open until August 6th this year, which was pretty crazy. So now I'm gonna just talk about the history of my study area. So historically, this area is volcanically active, and this is evident by the various volcanic domes and soils that are scattered throughout my study area. Geologists approximate that between 900 and 640 years ago, my study area erupted, and according to a Daily Mail article, my study area might be set to blow due to small earthquakes they've been detecting. So cross your fingers that doesn't happen while I'm doing my research. And then also historically, parts of my study area were grazed. In the 1950s, mid-1950s, it was heavily grazed by sheep, and the National Forest would allow about 2,000 sheep in my meadows for about 6 to 10 days. And now if we jump forward a few decades, the Owens River Headwater Wilderness Area, which is the wilderness area that occurs in my study site, was designated as a wilderness area on March 31st in 2009. I also want to acknowledge that where I'm doing my research is the ancestral land of the Tuolumne Band of Miwok Indians, the Western Mono, Southern Sierra Miwoks, Owens Valley Paiute, and the Mono Lake peoples. And then here's a list of notable historic and most prolific collectors. So the first specimen ever collected in my study area was by J.W. Condon in 1895. He only made a few collections in a creek called Dead Man Creek, which is a major creek in my study area. After Condon, there was a couple of decades where no one was collecting anything, but by the mid 1920s, F.W. Pearson did some minor collections. And then the infamous Mary D. Decker made a handful of collections in the late 1950s and then came back again in the mid 1970s. By the 1980s, Dean Taylor was another notable collector. And the most prolific collector in my study area right now is Helen M. Constantine, and she collected between 1994 and 1997. She is currently the most prolific collector because she conducted a study of the floristic affinities of the San Joaquin roadless areas. So her area focused on the western side of my study area, and she focused more on the headwaters of the Owens River. And the most recent collector, aside from myself, is Ann Howell, and she made collections between 2014 and 2017. And this is because she is currently writing a Flora for Mono County. So my study area is completely located within the Inyo National Forest and is managed by the Inyo National Forest. And because it is Forest Service land, they have a duty to the people and to protect the forest. So this means not land can be used as resources. And where my study area is, there's a lot of recreational activities. So this will include hiking, mountain biking, camping, fishing, hunting, OHV use, and in the winters, there's a lot of snowmobiling, cross country, and backcountry skiing. And this is all because Mammoth Lakes and June Lakes are also a big hub for outdoorsy types of people. And then timber harvest has and still does occur on small scales in my study area. People can collect fallen trees for firewood, but you cannot go and cut down trees. And then small scale mining has occurred sporadically in my study area, and there's currently a small active mine. And then, as I stated before, my study area was heavily grazed by sheep, but currently at a meadow called Dinks Meadow in my study area, it is used for horse grazing later in the summer when the meadow tends to dry out. So now I'm just going to talk a little bit about my methods. So my methods for this research include 2 field seasons, the summers of 2023 and 2024. Whenever possible, I will collect voucher specimens in triplicate to later deposit them at Rancho Santa Ana, the Jepson, and the California Academy of Science herbaria. The specimens that will not be collected in triplicate is rare plants if the population sizes are too small, and no physical specimens will be collected of federally endangered species. So Pinus albicollis, commonly known as whitebark pine, is a federally endangered species currently protected under the Endangered Species Act. And this occurs within my study area, so I will only be taking photo vouchers of this plant. And prior and during my field seasons, I use aerial imagery and maps like Google Maps and a Gaia, it's a GPS map thing, to check for interesting vegetation habitats that occur, and also check the topology to see what is accessible and how I can get there if it's not easily accessible. And before I did any of my field work, I created a species checklist by using relevant checklists of species in the area, and also I use California Consortium of Herbaria data. And I am currently in kind of behind on uploading all of my pictures of collections onto iNaturalist to make it more accessible to the public. So not only does my study area allow for a wide array of plant diversity, but it also supports various wildlife species. As you can see, I am in bear country. I have encountered bear while in the field. These are bear tracks I found in a meadow called Glass Creek Meadow. The Yosemite toad, this is a federally endangered toad that its critical habitat is in Glass Creek Meadow, which is a meadow in my study area. In these meadows, I find a lot of nests from ground nesting birds. And then there's trout and some brown trout. Unfortunately, living trout is too fast to take pictures of, so I only have a picture of a dead fish. And now I'm going to take you on a tour of the habitats that occur in my study area and at what elevations they occur at. So we will begin at the lowest elevation in my study area, and these are the lower, what I call the lower elevation meadows, and they occur from 7,300 feet to about 8,000 feet. And these meadows are very wet at the beginning of the season with all the snow melt, and towards the end of the season, they tend to dry out. And these meadows are so dense, you can't help but step on many plants as you're trying to collect just a few. Here you can find many species of grass and carex, but also some very cute plants, including
Rachel, can you hear me? Your audio is cutting out, but I don't know if it's just for me. Can anyone else confirm?
is it working now?
Thank you.
Better, yes.
Yeah.
Okay. And then relatively in the same elevation, we have some open Artemisia and Prunus habitats, and these take place where the mixed conifer forest opens up, and these habitats are more exposed, and they are dominated by small shrubs like Artemisia tridentata and Prunus emarginata. And among the shrubs, you can find some cuties like the Cymopteris, Terebithius, the Wyethiomolis, and other small annuals, including Calcordis, Diplocus, and some Ipomopsis species. Some of my favorite habitats were these riparian habitats that would occur at 7,500 feet all the way up to 9,000 feet. So yeah, these habitats I found at lower elevations, but also in the subalpine, which was very interesting. And these habitats are characterized by very wet soils with running water and are covered by mixed conifer forest canopies. These habitats, they would bloom all season for me, and they had some very charismatic flowers, including Lilium kilianum, Platanthera dialata, the Polemonium occidentale, but also not pictured Delphinium glaucum, and then various lupinus species. Now moving on to the mixed conifer forest. Mixed conifer forest is the habitat with the widest elevational range in my study area, and it occurs from about 7,500 feet to 9,500 feet, so it gets up to just about alpine. And most of these forests are dominated by Pinus jeffreyi, but there's also a lot of Abies magnifica, Concolor, and Tsuga martensia, and plenty of other pine species. But as the canopy opens up, you'll find the very exotic Eriogonum nudum, but also some very cute Calcordis bruneus. And pumice flaps can be found at 8,000 feet scattered across my study area. These habitats are mostly dominated by Eriogonum undulatum, Caloptridium undulatum, Linanthus pungens, and early in the season, they are dominated by Diplocus torii, which makes it look like a sea of purple when you're hiking through, which is really cool. And as we transition into the subalpine, we get to the high elevation meadows, which is at about 8,500 feet to 9,000 feet. And these meadows hold much of the plant diversity that is in my study area. You can't really tell, but the background picture is of Glass Creek Meadow, which is the longest intact meadow in the Sierra Nevada, and it is about a mile long. And then here are some annuals that you'll find in this meadow. So you'll find some ranunculus, a lot of ranunculus species. You'll find a lot of Erythranthe primuloides. I have found 2 or 3 pedicularis species. And then these meadows also hold the most diversity for my carex, and this meadow specifically holds quite a few of my rare carex species that occur within my study area. And then I also have 2 higher elevation lakes, and those are Yost and Fern Lake. And they occur at 9,000 feet. In the soil surrounding these lakes are mostly sandy. However, they do transition into some wetland meadow type habitats. And then surrounding that is stands of Pinus jeffreyi, Abies magnifica, and then you'll also find the occasional Pinus albicollis in these areas. And interestingly, this is the only place where I found this Gentiana calicosa, but I also find a lot of Cirsium andersonii at these elevations, and then some very cute astragalus as well. And as we transition out of the meadows, we are walking up onto White Wing Mountain, which is a mountain that occurs at 9,000 to 10,000 feet, and it is composed of loose decomposed granite. And we are transitioning into more of the alpine habitat. So as you climb above the tree line, I was able to find species like the Lewisia pygmaea, the Chenactis, and interestingly enough, I found this Menzelia, which is typically a low elevation intermountain species, but it was on the top of White Wing Mountain. And then here is San Joaquin Ridge, which is the most western edge of my study area, and the elevation here is anywhere between 10,000 and 11,600 feet. And this is where we get into true alpine habitat. Here, snow persisted all summer long, which was very fun and very interesting to climb over. And the plants up here get a little bit harder to find, but they get smaller and harder to find, but they're really cute. I found a lot of astragalus species, mostly Astragalus whitneyi. I found this Ranunculus scholzii, and my favorite find in the alpine so far is this Castanea nana. This part of my study area is also where I find the most Pinus albicollis. So after my first field season, I collected 1,016 vouchered specimens. I was out in the field for over a 4-month period, and I spent about 55 days out in the field. The black dots on the map indicate where I made all of my collections over the past field season. And then over this field season, about 900 iNaturalist observations were made in my study area, which is really exciting. People are getting out there and they're seeing what's growing and what's blooming. And I use iNaturalist in conjunction with my research for citizen science, but also so that I can check phenology to see what is blooming and where people are finding it. For next season, I'm going to prioritize areas that I didn't get to. As you can see on this map, there's quite a few areas that don't have collections, and I need to get there and see what's growing. I will also prioritize target plant species, and this could include rare plants, historic collections, and/or species that I did not collect that are on my species checklist. I also want to spend more time in the field next season, so I want to start earlier and end later so that I can ensure that I did a thorough inventory of my flora. And currently, from all of my collections and iNaturalist observations, I have 50 families have been identified, and my top 4 are Asteraceae, Cyperaceae, Poaceae, and Fabaceae. I have identified 152 different genera, and the top 4 genera I currently have are Carex, Lupinus, Eriogonum, and Poa. And I don't know about you, but these top families and top genera do not come as any surprise to me. And then currently I have identified approximately 193 taxa from my collections. However, as I identify more taxa, the number of taxa and genera will change and will increase. And if any of you are particularly interested or good at identifying Bocara or Carex, please let me know. I'm more than willing to share the fun of keying out those species. And now I'm just going to share with you my notable collections and some of my most favorite collections. So we're going to start with Lupinus duranii, also known as the Mono Lake lupin or the mono lupin. This lupin is endemic to Mono County and more specifically endemic to these pumice soils. And it has a CNPS ranking of 1B.2, meaning that it is rare and threatened. It has a NatureServe ranking of a G2, so it's globally imperiled. And then it also has a state ranking of S2, so it is imperiled in the state as well. Next one of my favorites is Frutillaria pinetorum, also known as the pine frutillaria. It has a CNPS ranking of 4.3, so it is a pretty secure species within California. And again, it has a global ranking of G4, meaning it's very secure, but it has a state ranking of S4, meaning that it is uncommon. And these plants are actually very hard for me to find. Every collection I made is because someone else found them, not me, which is kind of funny. And then one of my favorites is Pinus albicollis, also known as the whitebark pine. It does not have a CNPS ranking, but it has a global ranking of a G3, meaning that it is very vulnerable. And this plant was actually very recently federally listed as of December 15th, 2022, as a threatened species under the Endangered Species Act. And I am currently helping and working with the Inyo National Forest botanists by surveying for blister rust on these pine species, which is a fungal disease that is killing populations of whitebark pine within my study area. And then by far my favorite is Botrychium simplex. It's known as the little grape fern. It is a G5, so it is globally secured. However, within the Inyo National Forest and the U.S. Fish and Wildlife Service, it is a special status plant species. And this is because it is a great indicator for other rare species and other rare Botrychium species. And then here are not all, but most of my field assistants. I could not have done this work without them. They made my work so enjoyable and so memorable, and I just want to thank them. And again, here's a list of all my field assistants, not particularly in order. However, I did have to put my dad first. And then I also want to say a special thank you to Mayor, my advisor, my family and my friends, and all my funding sources for supporting me through supporting my research. Stop share.
Questions. I have a question. Since last year was such a heavy rain year, were there things that you found that like may not have been seen for a long time, that may have been dormant, or things that popped up that you did not expect?
I wish I would have started like this program 2 years ago, because I feel like I could have answered that question a lot better, but I did find things more abundantly than like the herbarium records have, or like CCH2, or even the Jepson had. Like I just found a lot more of everything happening. And even talking to the people in the area, they're like, I haven't seen blooms like this in years. So yeah.
That's good. Hopefully this year will be really good too. We're already getting a lot of rain.
Yeah, apparently we're supposed to be getting another cold winter up there, so we'll see.
Yeah, fingers crossed. Ron has a question in the chat. He's asking if your checklist is published on CCH2 or anywhere else.
No, it's not published on CCH2. I will. As of right now, I just have it private and it's shared between my advisor and I, but whenever all my collections that I've made and put into CCH2, they are there. So if you look at CCH2 data, they're going to be the triangles, not the circles, because they're just observations right now.
Okay, I have another question. I know you kind of go across a couple of different like habitat types, but in the ones that have somewhat of a fire history, I was curious when like maybe the last fire was, and maybe are there any certain like fire followers that potentially could be present, just haven't really germinated or grown if it hasn't right, burned in a while or something. I'm curious.
It hasn't.
Yeah.
Oh yeah, it hasn't burned in a while. I can't remember off the top of my head, but I think the last burn was like 2006, but it was very small. Because Crestview, it's a fire station, is literally right across the street from my study area, but things outside of my study area have burned recently, and especially in Yosemite, the Yosemite Valley is pretty bad, as we know. So I think it's a case of not if, but when it burns.
Mhm.
So I haven't seen any fire followers just because it hasn't burned in a long time.
Yeah, that'll be interesting. Let me think of another one. I feel like I've always have so many questions, but some of them are like dumb questions I never asked them.
Oh, please. There's no dumb
Wait.
questions.
Okay, so this year there was so much snow. Did that delay your start of surveying? Or
Yeah,
did
by
you stay?
almost a full month. So usually things start blooming in May, but I really didn't start going out till the end of May, beginning of June.
So is that logistically a problem for your team to, because you put them together in January and or something and they can't start?
Um, so the team is usually me, and then the field assistant, I kind of send out an email, or actually a lot of my field assistants were like,
Oh.
hey, I'm in the area, or they're helping another student at the garden, and I was like, oh, I want to keep doing this work. So it was just a very informal way of inviting people. I got a lot of text messages, a lot of emails being like, hey, I want to come out, and I'll be like, oh, I'm going up at this time, you can come with me. So it did throw me off a little bit at the beginning, but not too much. Once I got in the swing of things, it was just fine.
Yeah, when you get someone like Kira Moore saying, can I help you? Sure,
Yeah.
I'm ready.
Yeah, that was funny. He just happened to be studying or camping in my study area when I was there. So I was like, do you want to come collect plants with me?
Perfect.
So that's how I got there.
Laura had a question. Could you expand on Brad's question on good camping areas in the study area?
Yay, Laura.
Yeah, so I use like Gaia, which is a GPS map, and within Inyo National Forest, it's really nice because it's a lot of dispersed camping. So if you take Mammoth Scenic Loop, you can basically drive any Forest Service road, and if there's a turnout, you can camp there. So my favorite spots are up Dead Man Creek Road. And if you drive up far enough, the road eventually ends, but it's like a circle, so it turns you back around, so you can't really get lost. If there are forest road markers and you can drive down those, and I can't even remember them off the top of my head, you kind of just have to like take some time to explore. But Dead Man Creek Road, you can camp next to the creek, which is magical.
Oh, this is for like all-wheel drive or four-wheel drive? Could you go in a Prius by any chance?
I've seen them up there, and I'm impressed every time I've seen them up there. Yeah,
I'll have
I've
to
seen,
test it out then.
yeah, and the Teslas too. So
Oh gosh,
you can. I would not recommend it.
imagine.
Yeah.
Paul Doyle was asking, are there invasive species present? And then I will piggyback on that and ask, I know there were a lot of land use history of like grazing out there. Do you think the grazers, significantly impacted the system, or still like legacy effects of invasion in certain areas?
Yeah, so I have a ton of Bromus tectorum, and
Ugh.
then I get a lot of little invasive brassicas as well that are everywhere within my study site. I was recently reading a paper saying that the sheep grazing, you can still see effects because of all the Bromus tectorum, but also the veratrum that occur within those meadows. I don't know if I know they're native to California, but I don't know if they were native to this area. So yeah, there's still effects happening from grazing that happened. And then in the meadow that is currently used as horse grazing meadow. There's a lot of trampling. The soil is more compact. So yeah, I still see the effects of grazing.
So in an old lab I used to work in, we were investigating a potential biocontrol for some bromus grasses. And when we would survey in the Angeles National Forest, Bromus tictorum was heavily infected by a smut fungus, but for some reason it still persisted in the area. Have you seen a lot of Bromus tictorum infected by any fungi up there?
No, there's a fungus, like the blister or blister rust, that's affecting the Pinus albicollis. It has to jump from ribes or something else to
Oh,
the
wow.
pine trees. So I see rust on like ribes and some bocras and stuff like that, but nothing on bromus. Within the Inyo National Forest, I have their invasive species like plant list, and it's like ranked high, but it's like in a category where it's like, oh, we know it's here, but we really can't do anything about it. Like it's not a priority to eradicate, which is sad but understandable.
That's interesting though about the ribes and the white bark. Is it white bark pine?
Yeah.
I never heard of that before, but the rust fungus.
Yeah, it has to jump. It has to jump.
Is it blowing in the wind then, or is it?
I don't know.
Is there like a proximity that's required?
I assume when I would find the rust or a rust on ribes or something, I would look at the trees, and I wasn't like I was at too low of elevation for Pinus albicollis to occur. But also the blister rust is like this tiny, and it's like an orange dot that you can only see when it's like in its reproductive phase. So it's really hard to identify and really hard to see.
Could be birds. The bird's just feeding on the. I don't know if the bird would want to go up that high, but who knows? Maybe feeding on the ribes and then just hanging out on the trees, moving spores around.
They also suspect that the tree has to be damaged in some sort of way. So white bark pine is being hit by bark beetles, and now this blister rust. So when I was looking on trees, I had to look. Well,
congratulations, I guess.
I was looking for burrow holes from beetles and or birds that have already like damaged the tree or stressed the tree out in some way.
Interesting.
Yeah.
Yeah, something similar is happening it's our big berry manzanita. Is it Arctostaphylus glauca? So in some areas in like Santa Barbara County, there's some sort of fungus that's infecting and killing them, but it only enters the system if the plant is wounded. So if you like trim the plant with unclean shears, or they're like cutting it on the side of a road, it opens it up for infection. So yeah,
it's very interesting.
Yeah, are we just about nothing else in the chat? Any more in-depth questions, Sunny?
I could go on forever, but no, I'm good.
Then I think we will call it a night. Thank you, Rachel. You're still here. It looks like Laurel decided to celebrate a birthday or something, but that's all good too.
He's like a balloon just started floating behind her head.
So for those of us here, one more thank you, whether it's out front like this or you have some emoticons or something. There we go. Thank you. A nice and informative night.
Yay.
Anything else from you, Sunny? Are we just?
I will say, yeah, we are going to be starting putting a call out for O'Neill grant applications this year for the 24 year, and applications will be due on the 4th Monday of January, which I believe is the 22nd.
The next cycle. Good.
Nice cycle.
Get some more good speakers like this, like tonight.
Definitely.
Okay. Thank you again, Rachel, and everyone out there. Nice to see you. Happy holidays. See you in the new year. Bye.
Good night. Happy solstice.
Yes. Yes. Very good.